Yes.
Great. Thanks,
Okay.
Conrad.
Yeah, thank you very much. Thank you for the introduction. I'm very honored and grateful to have the opportunity to talk to you today, to be a guest at your journal club setting at Pan Israel. Yeah, it's such an honor. And I have a very interesting study for you. And yeah, as it was said, we talk about the Mediterranean diet and how it alters the microbiome in older people, which might has positive effects on frailty and improving their health status. Yeah, let's take a look at the study. It's a study published first in February of 2020 in Gut, a BMG journal. The first author being Tarini Shaka Ghosh and senior author being Paul W. O'Toole from Ireland. And I got this paper option from Zah, who, thanks to you again, who helped me very much with finding the right article and yeah, establishing a journal club. And I think why Zah chose it is a quite big study. It's a randomized controlled trial. It's the journal has a high impact factor, and yeah, the results are quite fascinating, as you might see in the next few minutes. Okay, let's take a look on the background of this study. As it was said, we look at frailty. Frailty is a complex problem of aging in human. It is characterized by mechanisms like chronic low-grade inflammation, loss of cognitive function, sarcopenia, and it is as a syndrome defined by symptoms like weight loss, exhaustion, low physical activity, slowness, weakness. And it's suggested that diet and dietary interventions may play a major role in the therapy of this complex syndrome. And this is why a restrictive diet and a low diversity microbiome is a challenge in elderly healthcare, as we all might have experienced already. And the Mediterranean diet as a popular plant-based dietary pattern and its adherence is linked to reduced frailty, and furthermore, frailty co-varies with specific alterations in the gut microbiota. And on this background, the New Age study was conducted. It is a multicentric randomized controlled trial investigating the effect of a customized Mediterranean diet intervention for 12 months on parameters of aging, 1,200 participants, 65 until 79 years of age, non or pre-frail, as I've said, multi-centric, based in 5 different EU countries: Poland, Netherlands, UK, France, and Italy. There was a baseline interview and a food record for 2 weeks for both groups, and whereas the control group got introduced into the general dietary guidelines, the intervention group got a specific training in the New Age diet. And the New Age diet, as we've said, is a specific Mediterranean diet, and it's tailored, modified to for the elderly cohort. And the intervention group got more support. They got free food like olive oil. They had 2 professional counseling sessions and monthly follow-up calls. Basically, data were collected at baseline and post-intervention. We measured dietary adherence and cognitive tests. This was the primary study, and now we take a look on the study we focus on today, which took a subset of these new age trial participants, more or less half of the cohort from all the different countries. There were additional data collected. We have gut microbiome profiles from stool samples, and we have additional biomarkers of aging and frailty. And the objective of this study was to measure the effect of the intervention on dietary pattern, the gut microbiome, and biomarkers of aging and frailty, and investigating the role of the microbiome in the development of frailty. And I'm very happy to show with you now the results of the study. First, we look at the dietary patterns. Yeah, I learned a lot. I want to say that I learned a lot about statistics while preparing for this journal club. And I've learned that this is called principal component analysis. And we use this method to summarize huge amounts of information by using summary indices. And when we take a closer look at figure A, which shows us the baseline dietary profiles, the baseline dietary pattern of all participants separated by country, we recognize that one data point stands for one participant. And the basic finding is that the baseline dietary pattern differed mainly by looking at the 5 different countries. Predictable finding. Different countries have different cultures. Yeah, but quite relevant when we look at this multi-centered setting. Figure B shows us now the microbiome pattern. So like figure A, every data points represents one sample of one subject. And here we see that there are also country-specific pattern in the microbiome profiles, and the authors made some computations and found that these changes in microbiome profiles are related to the changes in the dietary pattern. Okay, now let's take a look on figure C. Here we see likely the same like we have seen in figure A. Now we separate between intervention and control group, and we see that the intervention worked. The diet was different, and how different was it? This is what figure D wants to tell us. I don't know if this is readable at your screen, but it's not necessary because this, what figure D wants to show us is which food components drove this heterogeneity. And it was found that the intervention group ate more micronutrients and more fiber, micronutrients like vitamin C, vitamin B, copper, magnesium, and the control group ate more fats. So, and this was what drove the data points towards the right upper corner. So yeah, we might make a first conclusion here. The diet and microbiome profiles separate and co-vary by countries, and the intervention altered macronutrient profiles. This is a message the authors gave us, and now we take a look on the specific microbiome changes correlated with the New Age dietary adherence. And here we take a look on this figure B, and this shows us, I must give a little introduction here. The authors analyzed the stool samples and identified 130 OTUs, operational taxonomic units, aka species, and they did that looking at the 16S rRNA gene, which is a standard operation. And what they found is that they took the OTUs, the species with the highest, with the biggest responsiveness to the intervention diet. And on the left side, we see the diet positive group. These had a positive association with the adherence score. So the higher the adherence to the intervention diet, the higher the abundance of these species. And the opposite is true for that group. So, and these are tagged diet negative. And when we look at the names of these taxa, we see that these are not unknown species, but these species are associated with health, and these species are more associated with adverse health effects. So the authors conclude that increasing adherence to the New Age Med diet induces beneficial changes of the gut microbiome. And now let's take a look on this next slide. And now we focus on frailty and its associated biomarkers. And let me give you some introduction here. The higher the frailty score, the higher the abundance of the negative taxa, right? We saw these diet negative species. The higher the frailty score, the higher the abundance of the negative taxa. And we see that the diet positive taxa were more abundant in the stool of non-frail healthy subjects. With that being said, we must admit, though, that there were no direct association observed between dietary adherence and frailty score changes. This brought the authors to the idea that the microbiome plays maybe a bigger role than the diet itself. And now let's take a look on this heat map, figure A. At first, at the right, on the right side, we see the diet responsive taxa, the diet responsive species, sorted by their correlation with dietary adherence, sorted from low association, negative association, diet negative, to high positive, diet positive. On the horizontal, here we have the different biomarkers. Here we have the more negative biomarkers like interleukin 17, high sensitivity C-reactive protein. And here we have the more beneficial biomarkers like hand grip strength, and here this memory test, this cognitive test. We have adiponectin here as well. And the color code represents the tendency of correlation, or the trend of correlation. Red suggesting a negative correlation and blue suggesting a positive correlation. And if we look on this closely, then we see a basic pattern that the positive correlations are here and here, and we have negative correlations mainly here and here. And that means that, we have positive correlations between the diet negative OTUs and the bad biomarkers, and we have positive correlations between the good biomarkers and the diet positive OTUs, and vice versa. And what figure B now adds is that this pattern could be replicated also in the country-specific cohorts. So what the authors suggest now is that the microbiome changes associated with Mediterranean diet adherence are relatively consistent across the countries and are negatively associated with biomarkers of aging and frailty. Okay, let's take a look on that as well. Now we investigate more the question if the microbiome is maybe the missing link, and we test the hypotheses if that it's not the diet which links frailty and diet, but it's the microbiome changes associated with the diet. And therefore, the authors computed a microbiome score, which should summarize the microbiome changes in response to the Mediterranean diet adherence or to the intervention diet adherence. And they tested these correlations between the they tested the correlation between the biomarkers and this microbiome index. And this is the result of this correlation tests are here. We see on the x-axis the Spearman-Rowe correlation, and on the y-axis the negative decadic logarithm of the p-value. So the higher the data point, the more likely it is that it's statistically significant, and the more left it is, it appears the higher is the negative association with it, with the negative association between the biomarker and the microbiome index, and the opposite for right. And the basic finding is that 10 out of the 11 biomarkers, here we have 11 biomarkers, which where significant results were found. And here we have 10 of these biomarkers, which could be replicated, where the association could be replicated. And the authors' result here is now that they say the Mediterranean microbiome index correlates with reduced frailty, improved cognitive functions, and reduced inflammation independent of the adherence scores. Independent of the adherence scores, because they used by computing this, they used the dietary adherence as a confounding variable. And yeah, this is quite astounding from my perspective. Let's take a look to complete the picture. Let's take a quick look on the last result slide. Here we answer the question where the diet positive and diet negative species are located in this highly dynamic network we call gut microbiome. And yeah, this is also my first contact with network analysis. But what I've learned is that every node is a species, and every connection represents a core occurrence relationship. So when species A and B are connected, that means that they appear, the likelihood that species B will appear is higher when species A is also there. So they are like friends, if you like that idea. And green are the diet positive species, red are the diet negative species, and black the diet unresponsive species. And the basic finding is that the diet positive species are located more at the center, and the diet negative, and as we now know, frailty associated bacteria or species are located more in the periphery of the network. And this is also replicated by these box plots showing the degree centrality here and the between centrality of the diet positive and diet negative taxa. So what have we learned from this? Bacterial taxa that respond positively to Mediterranean diet intervention occupy keystone interaction nodes, and the diet negative taxa are more in the periphery. Okay, conclusion. As we've said, diet and microbiome profiles co-vary and differ between countries. Increasing adherence to the New Age Med diet induces beneficial changes of the gut microbiome. These changes are negatively associated with biomarkers of aging and frailty, and they are key intermediate between dietary adherence and health. Number 5, as we have seen it, let's take a closer look at the things we can discuss about this paper. I'm sorry, I'm a little bit over time. I will quickly add these thoughts here. How to put these results in the context of what we already know or what is already known in the field of microbiome research. The study suggests that the microbiome is the missing link maybe between the dietary adherence and the onset of frailty. And it shows that, as we've seen on the results 5 slide, that diet positive taxa have a keystone position in the dietary networks, and this supports the Anna Karenina principle of microbiomes, which states that microbiomes of healthy individuals are similar and the microbiomes of unhealthy individuals differ in their own specific way. Next point. Beneficial effects of Mediterranean diet interventions on microbiome level has been shown by other studies as well. So this correlates with the results I've shown you. And the authors suggest also that these identified diet positive taxa may be candidates for a probiotic therapy with the specific target to improve the health of frail people. Let's take a look at the limitations of this study. We have a low signal to noise ratio measuring the microbiome changes because of the baseline heterogeneity of the stool samples. So this is what I've shown you on the first results slide. The microbiome of people from 5 different countries differ very much, and this makes it harder to detect signals or responses. Yeah, they applied a novel machine learning methodology. Yeah, this new approaches are maybe there's not so much experience with new approaches, so this might be a limitation as well, but might also be an opening for more and better research in the future. We have the basic contradiction about the congruence of mucosal versus stool microbiome. I think this is a whole, this is a big topic in this whole field, that the mucosal microbiome and the stool microbiome may differ. But we only measure mostly the stool microbiome. And the authors also say that the global feasibility of a MET diet is limited by food availability, and that there are possible contraindications of a MET diet in the elderly, like dental diseases or dysphagia, which might make it harder to digest plant foods, or to digest plant foods. Yeah, with that being said, sorry for being a little bit too late. Okay. Yeah,
thank you so much.
And